Let's now take a look at the sympathetic or adrenergic physiology and pharmacology, and we're going to start with not only the signal transduction from the standpoint of synaptic transmission, so we understand the way that happens and how drugs may be impacting it, but also to understand what's happening at the end effector tissue. And this is where these are the signal transduction mechanisms that are you've heard about already in the parasympathetic nervous system that are in part the way that things get integrated at the cellular level through inputs of both arms of the autonomic nervous system. Now the focus of looking at this slide is not to look at anatomy as a key element here, because I really do want to focus to the clinical effects at each of the tissues, but I do want to point out that we call the sympathetic nervous system as the thoracolumbar outflow, and so you can see that most of the interactions coming out of the spinal cord are via this region of the body, and in that we have ganglia that sort of coordinate things from there. We also have a long pathway from these ganglia to the tissue, and so this is a system that is set up to lead to rapid outflow or diffuse activation of the body under the influence of the hypothalamic control of the autonomic nervous system. Now in this slide I'm trying to, in one cartoon, to kind of represent the prototypical synaptic transmission between a presynaptic neuron here and the end effector cell postsynaptically. Now it could be another neuron, it could be any tissue that we're going to talk about. And let's focus on because it's simpler. We will be talking about drugs and sites that are hitting on receptors for norepinephrine or even epinephrine, the alpha 1 and beta 1, 2, and 3 receptors, depending on the tissue. Rarely does a tissue have all of these. And the other possibility is the dopamine receptor. So this is dopamine. And again, not always on the same tissue, in fact, usually not, but basically the point is the effects in the drugs are focused on understanding where those receptors are. If you go back to the table in one of the earlier units, you'll see there's a list of where these receptors are, and we'll review some of it as we go through here. But I want you to understand how the signal transduction works. Now, on the presynaptic side, so this is our synaptic cleft here. We have the release of, let's say, either norepinephrine denoted in pink, or right here, or we have the release of dopamine here. And let's just focus on the fact that one or the other is being released. So it makes the drug makes its way across, it finds its receptor, it's reversibly bound, signal transduction, which we'll talk about, occurs. And well, how does this signal end? Well, the signal ends in most cases by a protein that is just outside the synaptic cleft that picks up all the extra norepinephrine. Let's start with that. And there's a norepinephrine transporter that pulls it back into the cell and it recycles it. And so basically, signal is terminated by reuptake. There's possibilities, which we won't go into in great detail at least, that allow then for there to be metabolism to occur, and we do have drugs that influence that metabolism usually to inhibit it. So, and this general motif is true for a catecholinergic presynaptic neuron. So in the case of dopamine, it's dopamine being released, and it's being taken back up by a dopamine transporter. Now, the other thing we see is that there's a location of on the presynaptic neuron of some of the norepinephrine or catecholaminergic receptors, the alpha-2 receptors. And these are there to sort of serve as both the way another neuron might impact the control of this neuron that's secreting epinephrine. And we'll talk about drugs that actually target alpha-2 receptors. But it also serves as sort of a negative feedback on the system itself. So it creates a little braking mechanism by having this alpha-2 receptor being linked to a reduction of additional transmitter being released. So we have a number of possible mechanisms where drugs can have their effects. But understanding where these receptors are and the subtypes, depending on the tissue, is very crucial. And that's where we're going to go from here. Although we alluded to this in a prior cartoon, I basically want to just summarize, well, how does the action, once it's started, how does it end? Well, obviously, this is a reversible type of interaction with the receptor binding. So we can get dissociation and then that occurs more likely if the secretion or the release of that catecholamine is reduced. We get reuptake, we talked about that, the transporters, and then metabolism. And for the most part, these drugs, if you gave them into the plasma, they have very short half-lives. We do use dopamine and we do use epinephrine, sometimes given IV, but we know that their action is, we're kind of requiring it to be quick. We're requiring the action to be controllable by the metabolism. So we have a half-life only minutes long. And then the eventual degradation is by two enzymes called catechol-O-methyltransferase and monoamine oxidase. And in the latter case, you'll see there are drugs directed at this enzyme that are usually targeted to try to increase the local amount of a neurotransmitter, whether it be norepinephrine or more likely now dopamine in the synaptic cleft. So let's talk about the cellular mechanisms. And the reason, again, while I promise you clinical stuff here, I really want to focus mainly on showing you that here we go, the sympathetic nervous system is impacting intracellular signaling of calcium, of ion channels, of adenylate cyclase, etc., phospholipase C, just like the muscarinic or parasympathetic nervous system. So we said that understanding some of this signal transduction and how each subtype of receptor is linked to a slightly different mechanism, depending on the tissue, is actually really interesting to understand in terms of how all of this gets integrated at the cellular level as a particular cell might have input from both arms of the autonomic nervous system. A general concept that's well worth understanding is that all of these receptors are G protein coupled receptors, all of them. And so everything follows from that, the specificity of the G protein, what it's linked to in terms of signal transduction within the cell. So in this cartoon, I basically want to give you an example of the beta-1 receptor, let's say on the membrane of the heart, the cell membrane of the heart. And this receptor is linked through G proteins to the adenylate cyclase and enzyme. And so this is a motif we really hadn't talked about before. It's the only one we're going to talk about specific to signal transduction with regards to the adrenergic nervous system, because we've kind of already talked about the other ones, phospholipase C, protein, and protein kinase C. So here what happens is we get the stimulation of the production of cyclic AMP, and that activates protein kinase A, and that can have a couple of different effects. Either the more rapid effects, which we'll talk about here, for example, increase of heart rate and contractility, might be due, just very simply put, at least in part, by phosphorylating a calcium channel and the entry of calcium and the calcium-stimulated calcium release at the sarcoplasmic reticulum that you learn about in cardiac physiology. So that's a rapid effect. A slower effect is that these protein kinase A can impact CREB, which is a signal transducing protein that can bind to CREB binding protein on the nucleus, and that can lead to transcription and translation of proteins. And so, for example, if you have constant or chronic stimulation of the beta-1 receptor, you might get cardiac hypertrophy. That is an actual cellular response requiring the cell to make more protein.